Hi everyone, I'm Paula Saffi, an MD working in Chile, treating patients with Parkinson's disease and related disorders. I wanted to share today my experience during a sabbatical stay at NIH with the CARD team. We tried to use long-read sequencing to improve the research in movement disorders genetics in limited resources scenarios like Chile, where I live. I have no personal disclosure. So let's start with the problem. That's a problem that I see every day in my clinical practice. For most patients with suspected hereditary movement disorders, getting the diagnosis is long, is expensive, and often is unsuccessful. Even if you have access to gene panels or exome sequencing, many variants are missed, like repeat expansion or structural variants, leading to an odyssey or a very difficult journey for the patients. This New England Journal of Medicine study shows that genome sequencing improves the diagnostic yield, especially for structural and intronic variants, but also shows the inequities that underlie underrepresented countries and regions like Chile, where the diagnostic rates are much lower. That's where our work fits in trying to reduce the diagnostic gap, tailoring new technologies to solve complex genetic disorders. For this exciting and innovative pilot project, we selected 15 samples with high suspicion of an underlying genetic disorder. 4 of them were samples of Parkinson's or Parkinsonism patients, including early onset, atypical, and familial cases, and 6 of them were ataxic with different phenotypes, but all of them had prior negative genetic results. So our goal was simple but ambitious. Use one tool, which is long-read sequencing, to identify known pathogenic variants and investigate the genetics of those rare ones where no cause had been identified. With the support of the CARD team at NIH, first we developed a non-high molecular weight DNA extraction protocol. The reason was because this wasn't able to do in Chile because these are expensive and need a lot of technology. So we specifically designed a protocol that it was simple, low cost, and suitable to use in settings with limited resources like Chile. The protocol is now published, and you can access it by the QR code in the slides. After the extraction, each sample was run individually in one flow cell. And as you can see, the data quality was excellent. The average output was 133 gigabytes per flow cell with a 40x coverage and N50 around 22 kilobytes. Then we use a modular analysis pipeline to capture the different types of variants. We use Piper margin deep variant for single nucleotide variants, Sniffles for structural variants, and VAMOS for short tandem repeats. We also perform ancestry analysis, and you can see here in the black circle how our Chilean samples cluster with a mixed American population. That was something that we were expecting. Among the Parkinson samples, we found that 4 of them had pathogenic single nucleotide variants in 2 different genes. There were 2 samples that carried 2 different GBA1 variants, and 2 samples that carried the same G2019S LARC2 variant. These are well-known pathogenic PD variants and reinforce the power of long-read sequencing for this SNP detection. More interestingly, we identified the zinc finger homobiox gene repeat expansion in 4 samples, which is the underlying cause of spinocerebellar ataxia type 4, or SCA4 for short. And this is very important and an amazing discovery because until now, all these cases were reported from Northern Europe. So this is the first discovery from Latin American samples. In this table, you can see the clinical features of the 4 carriers. All of them had the previously reported features of cerebellar ataxia and sensory neuropathy. But some of them had other features as upper motor neuron involvement or even dysautonomic features, REM sleep behavior disorder, and dementia. There was one very interesting phenotype that consisted of a complex ataxia Parkinsonism spasticity phenotype that also had gaze palsy and myoclonus. This expands what we already know about SCA4. And in the right panel, you can see 2 MRIs from 2 different individuals. In the top one, you can see the Burmese atrophy and also spinal cord atrophy. And in the one at the bottom, you can see the cerebellar atrophy. Well, this slide shows individual allele plots for each of the 4 carriers. In blue are the repeat length distribution per allele, and you can see here that all 4 individuals have one expanded allele above the pathogenic threshold that is highlighted in the red line. In all of these samples, they had the same motif, that is the GGC repeat. We also explored the relation between repeat size and age of onset, and we saw that there was a negative trend with larger expansion leading to earlier symptoms, but it was statistically non-significant due to the small sample size. In these plots, we are showing the haplotype-specific differential methylation around the expansion. On the left panel, you can see an unaffected family member. In blue, you can see that the methylation across both haplotypes is low, which is what we expect under normal circumstances. And on the right, you can see 2 affected family members. And you can see in red, in the expanded allele, that is the haplotype 2, that is hypermethylated, while the non-expanded allele, that is the haplotype 1, remains hypomethylated. And this differential methylation pattern was reproduced across all 4 carriers. Together, this data not only confirms the allele-specific methylation, but also suggests differential regulation at this locus. While the functional impact remains to be fully understood, this provides a strong foundation for follow-up studies to investigate how methylation and this expansion may influence gene expression using transcriptomic approaches. Here, I'm showing that we also perform haplotype analysis to explore the origin of the expansion. Here we can see the genotypes across a panel of SNPs surrounding the expansion, and in the top row you can see the unaffected individual, then you can see the 4 Chilean samples, and at the bottom you can see the Utah index case from the Chen et al. Study. The pink shadow shows the 6 rare single nucleotide variants that were previously associated with the Northern European founder haplotype, and 3 of them were present in the affected individuals and were absent in the unaffected one. So this suggests a common founder effect for our samples, which is surprising because of the highly admixed population that is in our country, in Chile. So to summarize, we are happy to share that we created a low-cost, non-high molecular weight DNA extraction protocol that has high quality for long-read data in resource-limited settings. We got to use one single test to identify known pathogenic single nucleotide variants in Parkinson's disease genes, but also we got to identify with the same tool expansions in a gene that was only reported in Northern Europeans, which is SCA4. So with this pilot project, we expect to continue advancing the genetic understanding of underrepresented populations. I want to finish thanking with all my heart to the CAR team, especially Kimberly Billingsley, John Moller, and Kim Paquette for their support throughout this project, and to Nanopore for the opportunity to present this work. Thank you very much.